colloquium that he was initially invited out here for. I've known Russ since the year after his bachelor's at UC Davis, when we were both in Chuck Langley's lab there, working on some of the earlier genome resequencing of Drosophila melanogaster. And, within that post-bac year, Russ figured out how to basically call the inversions of Drosophila melanogaster from short-read sequence data, and took that with him and got a flying start to his PhD at Harvard in the lab of Dan Hartle, who, as I mentioned the other day, is a UW-Genetics alumnus. Subsequently, he did a postdoc with Rasmus Nielsen at Berkeley, and then jumped off to form his independent research lab, I think you said 8 years ago.
Yep.
And he's covered a really amazing span of projects in that time. Some of us heard about one yesterday that had to do, or actually a couple yesterday that had to do with the evolution of genome structure in terms of introns and inversions. And today we're going to hear about Russ's pandemic project, on a pretty enormous scale. And figuring out what to do with all that COVID sequence data and building better trees with it. All right, join me in welcoming Russ, please.
All right, well, welcome back. Thank you, John. So I'll introduce myself very quickly. I guess you kind of handled this yesterday as well. So in the pandemic, we got really interested in this problem of, we had almost infinite genome sequences. That's an exaggeration, obviously, but there are currently more than 16 million total available SARS-CoV-2 genome sequences, and basically no way to handle that. And this will be sort of a little bit about how we did that. So some methods and how we constructed these, some of the impacts, and then what else we were able to do with these data. Just to start with, I don't think this will be a particularly controversial statement for this audience, but phylogenetics has been really a central part of the effort to fight against SARS-CoV-2. So this is a screen grab from actually my competitors, but I really like their work, at nextstrain.org, where what I'm showing you is a phylogenetic tree. So the idea here is that samples that are more closely related will be these points that are sort of more close together here. They'll have a more recent common ancestor, that is, their branches will find each other. And in this case, they're colored by the region of the world they were sampled in. Right? So one thing you can see really easily, so blue is kind of Asia. If we go back here to the root, that's sort of the ancestor of SARS-CoV-2. Pretty clearly, the first major emergence of SARS-CoV-2 must have been in Asia, as far as we can tell. And then it subsequently spread into the rest of the world, first into Europe, back into Asia, those sort of blue streaks up there, and then in the Americas and other places as well. So this is one of the things that we can get out of phylogenetics, is a sort of overall depiction of how a virus is transmitting across global populations. There's obviously much more. Okay. And so the reason this works is that your transmission itself defines a tree, right? It is a tree. Where the first individual, patient 0, might transmit to 2, and they'll transmit to some more. Each of them transmits to some more. And then, if at the end of this, we're able to throw some genome sequencing on top of this, as our mutations accumulate over time, we can sort of indirectly go back and infer how the samples must be related to each other. So for example, these two share the orange mutation, so they must have a common ancestor before they join up with all the others. So that's the big idea here. This, of course, was incredibly popular, right? It became very easy to do this. It's sort of, the pandemic happened at kind of the right time in the genomics development, if you will. Wrong time for everything else, obviously. And so we could put sequencers almost everywhere in the world. Like literally, there are people in the bush in Australia sequencing the virus. There's people all over the place. And so it was a massive global effort. And there's a couple things you can do with this, sort of high-level ideas. One, you might want to do what's called viral surveillance. The big idea there is that if I see a new variant or a new strain that has emerged, can I figure out that it might be important? And in particular, it might drive a new wave of infection in my community and overwhelm my healthcare system. So for example, this is just from a screen grab from outbreak.info, one of our collaborators, showing the sort of waves of variants early in the pandemic, where initially there were tons of lineages, and then Alpha pretty much won in the UK. Delta came out of probably India shortly thereafter, and then Omicron took over like immediately after that. And because the WHO will not rename any more strains, we just have Omicron forever now, although there are new variants arising all the time. I know that's very confusing branding, but here we are. The other thing you might want to do with this is something a little bit more local. We sort of call it genomic contact tracing. or outbreak analysis, where the idea is that because I know the very, very fine-grained relationships among my samples, like I know these three, because they're all on this vertical line here, are genetically identical to each other. There's no mutations that distinguish them. They must share a very, very recent transmission history, right? It must have come from somebody relatively recently, because otherwise mutations would have accumulated in a sort of semi-clock-like fashion over time. So the classic phylogenetics idea. And so what you need to do this, in any case, is a pretty much up-to-date and comprehensive phylogeny. I'd like to have all of my information in one place. In the case of outbreak analysis, if I have these 5 samples from my community, I can make this tree, but I actually can't know on its own, was this one introduction into my community and subsequent spread? Was it actually several different introductions into my community? But if I have all the other information that's available to me and I project that on here, I might see something like this that tells me, okay, there may be some community spread, but it's very likely that at least there's 2 separate introductions because there's so many samples from elsewhere. It's unlikely that it's in my community and spreading everywhere else all the time simultaneously. More likely, it's very diverse coming in. Of course, this doesn't answer the question, it just gives us clues of one reasonable interpretation of this history. On the other hand, you might have something like this, where I have a whole bunch of samples, maybe 10, and I say, is this special? Is it a new lineage? Should I be tracking this thing in time because it might drive further waves of infection? And if I know about all the rest of the samples, again, I have the same sort of thing where, okay, that one, they really are really similar to each other and really different from everything else. Probably I should be tracking this particular wave. I'm going to label that and follow that in public health discourse. Okay, so what I'll talk about today, how we do this, how do we infer and maintain a comprehensive phylogenetic tree, a little bit of our work using this kind of inference to learn about recombination in SARS-CoV-2, and then finally, boldly titled, some speculation about what I think the future of pathogen online phylogenetics is going to look like, how we're going to use these things to positively shape societies. Okay, and so within this, there's sort of 2 sub-questions. The first is, how can we do it? Like, is there a good way to do this? And secondarily, I'll go through a couple examples of how this has been useful in public health and how we're seeing this applied in sort of real time as we're making this resource for everyone. Okay, and so here's the big problem. I should update this. It ends in 2022. There's been more data since then, but the scale and the pace of SARS-CoV-2 data is just extreme, right? Like if RSV, respiratory syncytial virus, had about 1,000 genome sequences before the pandemic, and flu made a couple, maybe 5,000 in a year or 2 before the pandemic. And then suddenly in a single week, we made 163,000 genome sequences across the world. You can imagine that most of our bioinformatic infrastructure just wasn't constructed for this problem, right? We were sort of retrospectively analyzing a few hundred or a few thousand sequences that sort of trickled in slowly. And instead now we have this deluge of data that we need to be able to grapple with. Okay. And so the big problem here is the way we do this. So the typical approach for using these data would be what we call de novo phylogenetics. Those of you that have made a tree, an IQ tree, neighbor joining trees, et cetera, that's de novo phylogenetics. I take all my data, I figure out how similar they are to each other, I infer a tree one time. But the way that would work is something like this. I gather all my data, I make my alignment matrix, I infer my tree. But tomorrow I have new data, right? And so my tree's already out of date immediately. And so what do I do? Well, I can append it to my data matrix and recalculate my tree and just start over, right? But you can kind of see where this is going. Obviously, I'm going to struggle because the next day I'll have even more data and I could do it again. But eventually, even just a few months into the pandemic, we knew this wasn't going to work, right? We were literally a few months in, there were all of 10,000 genome sequences, and it was a problem because it was taking us 3, 4, 5 days to make these tree builds. And so just extrapolating into the future, we knew we had a problem. Okay. And so what we came up with, and I'll freely admit, I don't think we're the only people that has invented online phylogenetics. It's an idea that's been around, like all great ideas in science. But the big idea is that's de novo phylogenetics. Well, what if instead of doing that, I do what's called online phylogenetics? And the idea here is that on day 1, I have to infer a tree. I got to start with something that's green. But on day 2, I have all these blue samples. I'm not going to make a new tree. I'm going to figure out how they're related to all the other ones. And on day 3, I'll do the same with my purple samples. And in that way, the tree can just sort of grow with the pandemic and follow along, if you will. And so the reason this works really well, so maybe day 3 is not that important, but day 100, I already have 99% of the data that I need to know about. I only have to do a little bit of work to figure out how the rest of that is related to it. And I can just sort of remember what I've already learned this whole time. And so it scales and scales and scales. Okay, so how do we do this? Like more technically. This is really work from my postdoc at the time, Yatish Sharakia. He's now an assistant professor at UCSD with a really incredible lab. Continuing work like this and other stuff. And so what we did was we said, well, we should think critically about our data structures. Like, how do I want to represent my data? What we came up with is what we call a mutation annotated tree. So this is just like a tree. It has sample tips A, B, C, D, just like your tree does. But what we're also doing is we're labeling every single branch with the mutations that we infer to have occurred on that branch. And the reason this is so useful is that imagine that instead of sample A, there's a million descendant samples of that node. I don't have to record that single mutation that occurs on that branch 6G a million times. I record it once. And as long as I know my tree, I can just propagate that down and say, I know this must exist in all those samples because of my tree. And so when you do this, this data structure, I'll show in a second, is just absolutely tiny. It's sufficiently small that it fits in what's called the last level cache of your CPU. And that's why you can be so fast to operate on these sorts of objects. And so when I want to add a new sample, I'm just going to use a type of maximum parsimony. We do have likelihood versions of this now, but parsimony is still what's in use because it scales to where we're at. So you can do, what you basically do is you say, I'm going to take my new sample and I'm going to compare it with just brute force. I'm going to try every single possible placement on the tree. And I'm going to pick the one that requires the fewest additional mutations. So in this case, you can see that mutation 10U is shared with branch 6. So I know that sample E should just slot in next to D, something like that. And I can just write back out what's called another mutation annotated tree. It's actually the same thing. I just appended a little bit more information on top of it. Okay. And so what's really great about this thing I was mentioning is evolutionary compression, is that I don't store a giant matrix of all the mutations that I've observed. I only store this little bit of mutations where I think they may have occurred on my tree and the tree itself. And so if you have a 50,000 sample mutation annotated tree, it's about 1.7 megabytes. The current one that's about 16 million genomes plus is about 350 megabytes. So I literally email this to people as a Google Drive link when I send to, which is very convenient for sharing all these data. Okay. And so finally, just to show you a little bit of this, this wouldn't be any good if it wasn't actually as fast as we hoped it was. And so when we got started on this, this is my student Brian, who's now gone on to a career in the industry. He said, well, this is an old problem. People have done phylogenetic placement. Like obviously somebody must have figured this out. And so he started profiling software. And so he tried pegging to IQtree and a few others. And what he found was that each of them, just to add 1,000 sequences to a 40,000 sample tree, took more than a day. So that's obviously pointless if I want to do online phylogenetics because I'll fall behind every single day and I'll never catch back up. And thankfully, when we finally got Usher to run, it took about 43 seconds for the same problem. So orders of magnitude faster. And we've actually made it much faster since then for reasons I won't go into today because they're basically hardcore CS stuff that I don't think is that interesting to present. But it has been made even faster and it's really, really grown nicely. Okay, it wouldn't matter if it was fast if it wasn't also pretty accurate. And so it does quite well with placements. And so when we test this, what we do is we either simulate a tree or we take a tree inferred by other software, we prune a sample out and we add it back in. We say, did we get the right spot, basically? And what we find when we do that is that 97% of them are topologically correct. That is, it goes exactly where it should go. And then in some rare cases, they're not quite correct, but it's typically about one node off, which would be, for example, user sample 1 joining with existing sample 2 or something like that. And so it's very nearby in tree space. When it's wrong, it's much more to do with missing data than it is anything else, as far as I can tell. And so it's not really a solvable problem on our end. And so we've moved on. And so this is an example where parsimony does really well, in part because there's just very, very few mutations. So the probability that there's more than one mutation on a branch is very, very little. And so it just isn't a big issue. Okay. So to tell you where we're at now, the current big tree, or big tree as we call it, is about 16 million samples. We update it every day. And each new edition takes about 4 seconds in total. So it's gotten a little longer, but it can hold up on our servers, no problem. We run it on local servers. And the entire pandemic, again, is encoded in a file that's 350 megabytes in size. So quite small. And just to show you, you can visualize this yourself. If you go to this website here, and I'll show you a little more in a second. This is cove2tree.org. It's from our friend at the Crick, Theo Sanderson. It's this beautiful interactive platform that will run entirely in your web browser where you can explore the full tree, all the mutations, everything else, and just kind of see how they're related to each other. And so we really enjoy this. And just to show you a little bit more, you can do this. So I'm getting kind of toward the application side of what you can do with the mutation annotated tree. One thing that we really love, this is work by my student, Alex Kramer here, is the treenome visualization. And so what we're doing here is we're taking that encoding, right? So the mutation annotated tree is an encoding of all the variation on this background that is the phylogeny. What we're doing is we're decoding that. So what that basically means is I'm descending the tree, I'm finding all the mutations in all the samples, I'm actually displaying those mutations as these colored lines relative to the root of the tree or the ancestor of this entire pandemic. And so you can also play with that on this platform. This is not super mobile optimized. So I've given you a QR code, but I really would recommend using a laptop or something else if you want to play with this. It will work on your phone, but only kind of. So maybe don't. But again, you can get to it from code3tree.org as well. Okay, some of the fun stuff you can do. So probably the biggest impact of this has been our integration in the UCSC Genome Browser, where what we're doing here is we've made a little portal where you can either paste your sequences or upload a file. And we'll just do the placement for our users. And so this is actually kind of the end state of a lot of public health offices workflow. They get their sequences back from their sequencing core, they upload them on here, and we'll display them for them by just adding to the tree and producing these visualizations for them very quickly, where I may parse it down to 50 samples in this case. I'm showing you, okay, that's how your samples are related, those three up there, implying maybe there is a single introduction or a relationship among the samples in your community. But you can also figure out from this pretty quickly. I don't know that you're be able to read the words on here. I apologize, but if you go up the tree, you can see this first sample in black here, which would be one of the ones we already knew about. So it can give you clues on where in the world new stuff in your community may have come from pretty quickly. And so we've used this quite a lot in California to sort of track international transmissions. When we see new stuff showing up, where is it coming from? And try to go through these sorts of visualizations and sort of automated tools to figure out what the major sources of transmission are into different areas. Okay. The other big impact. So this is now the basis for Pango lineage designation. So many of you may have heard the phrase like B117, for example, was one of the first we talked about. That has gone on. It is the main way that we track the virus now, is these kinds of approaches. And so what we're doing there is we're labeling big chunks of the tree, because the vast majority of public health isn't going to say, I have a sample that's like this one, but with these 4 mutations, or it's these, it's not a very useful system. Instead, you want some way to sort of a dictionary that tells me, okay, this whole block here is AY4. And so I've got a bunch of AY4s. And so, they have some epidemiological relationship to each other, where they may behave similarly across different communities. Okay. And just as an example of that, so the Omicron designation was made based on this, and you can actually look it up on GitHub if you're interested. The whole thing's done in a very open science framework, where we put everything on the internet kind of as soon as we know about it, and hope it's correct years later. This one held up, so we were all right there. And so that's kind of how this works. And then finally, I'll just show you a local application, sort of that outbreak analysis or local transmission analysis type thing that we were able to do in a collaboration with Marin County Public Health in California. And the way this works is, what I'm showing you is actually a seating chart. So they had a classroom, and they had a teacher who had symptoms, but refused to get tested and continued to teach for reasons. And so we won't go into that or comment about the logic of this. I'll say that they sat at the front. This is the seating chart of that classroom. The teacher's at the top there. And then across the front row, and then a little further back, there's all the symptomatic students and the positive cases, positive in blue here. And what you can see, I think, pretty easily is that the front of the room is a major risk factor. Like if you sit in those first 2 rows, you are really, really likely to get SARS-CoV-2. If you sit further back, you actually have a pretty good chance of escaping in this case. And so the question became, well, the teacher won't let us sample them for reasons. And there's a lot of diversity in the community. Is it just that all these kids got sick, or is it really that there's maybe one source of the infection that is subsequently spread through this classroom? And that's something phylogenetics can answer pretty quickly. And so the kind of transmission depiction you might have is something like this, where we have one individual infecting most of their classroom, and then they go on to infect their parents and siblings and people like that. Or is it many different sources? And in this cases, I'm showing you just a subset of the phylogenetic tree you get, but you can see that because they're all basically on this one line, those genomes are all genetically identical. This subset of related cases is one mutation off. And so the implication here is that actually, yes, this is very likely a single source of the infection. We can never demonstrably prove that it's this particular individual, but it seems very likely that we should be watching to control those major avenues of introduction into our classrooms and communities. Okay. Cool. Okay, so just to conclude from this first section before we go on here, so using Usher, we're able to maintain this completely up-to-date, pandemic scale phylogeny. I do think, I've been really pleased already to see all sorts of new things people have built on top of this idea. So there's a Bayesian framework from the Broad Institute that does some nice things. There's a maximum likelihood approximation version of this that's coming along pretty quickly. And so lots of different people have kind of built on this idea to build online tools to try to keep up with pandemics. And I think, I've been really excited to see what's coming out of this, because I do think it's the future of how we're going to do this, as I'll talk about a little at the end here. And then finally, this application of these tools lets us do really very, very fast and scalable outbreak investigations and lineage designations, right? So, you know, a classic rule of web development is 8 seconds or less, right? If it takes longer than that, people get bored. And so that's been very much a guiding principle of everything we're doing here. It's got to be fast, has to be displayed on your web browser, has to be something you can access immediately, and has continued to be part of this, although it does take more than 8 seconds. And I'm slightly embarrassed to admit, but we'll work on that, I promise. Okay, so cool. So with that, that's kind of what we're doing, why we're doing it. In the second part here, I'm going to tell you a little bit about some of the biological inference we're able to get out of these tools and kind of what we can learn about the evolution of SARS-CoV-2 by encoding this data and using this giant data structure to do this. And in particular, same sort of question here is, can we accurately detect recombinants in real time? Right? Is it possible for me to go in, look at a brand new sample and tell you, yes, no, this is a recombinant? And maybe more fundamentally, are there parts of the viral genome or different lineages where we see lots and lots of recombination, maybe indicating that this is an important source of epidemiological and novel variation, right? Is this something that's driving infections, or is it some weird thing that's happening in some small subset of viruses? Okay, and so to explain how this works, it's not the same as recombination as you may have learned it in typically eukaryotic organisms. This is a plus-stranded RNA virus. It's a very, very different thing that's going on here, but it's the same sort of thing that basically comes out of it. And so the way this works is, and of course, I've just spent all the time telling you how to make a tree. Now I'm going to tell you how it's not actually a tree at all, but that's okay. The way this works is you may be replicating your virus genome, right? So that's the RDRP, it's the polymerase scrolling along the virus genome, and it falls off for some reason. So it's just sitting here. And what it's able to do is find a homologous template. And as long as that template, in this case, the kind of yellow-orangish thing here, is genetically distinct from the original, you'll get a recombination where you get a genome that comes out that's a mosaic. It's sort of half this one, or wherever it splits. Okay. And so one thing we actually know from, this is much deeper time, like super deep time phylogenetic analysis here, but just looking at the relatives of SARS-CoV-2, that's beta coronaviruses, what I'm showing you here is different regions along the genome. So it's been subdivided by these colored blocks. Each of them has a different characteristic tree topology that relates the various samples to each other. And this strongly implies that recombination has been ongoing and is an important part of viral evolution on very, very deep time scales. So it's something we think might be important, even within human populations, although we don't know this yet. And in this case, we're actually tracing along these particular lineages to see how it moves along the topology, right? So that it's rearranging. Okay. One reason you might care, recombination will affect your phylogenetic inference, right? If your tree is not actually a tree, you won't get it right. And so it's important to start thinking about this a little bit for tracing the evolution and spread of the virus. And secondarily, and I think probably the most important reason I care, is that because you get these sort of multi-mutation changes, right? It's not just one mutation added to a haplotype, it's a mixture of two completely different viral genomes. You can get epidemiologically novel lineages. You might get a lineage that comes out of this that has very different properties to the two that went into it, and again, might drive major waves of infection across human populations. So we care quite a lot about finding these things. And as an example, there are many known now, but BA1 through 3, so these are the early Omicron lineages, are almost certainly complex recombinants where in the colored boxes along this top is kind of what we call a haplostrip. I'm plotting each and every variant that distinguishes these. And the colored boxes are cases that are just inconsistent with the tree topology, sort of the main topology that relates them to each other, strongly suggesting there's been an amount of recombination ongoing among these lineages. And this is actually some of the evidence for why we think Omicron probably evolved initially in an immunocompromised individual, generated lots of diversity, recombined a lot, and then several lineages came out more or less simultaneously. Okay. But before I get into how we're going to do it, I'll start by saying it's hard. So when you want to find recombination, the conditions for this, besides those individuals that are immunocompromised, are actually extremely conservative. It makes it very, very hard to find these things. And the reason for that is that you need your viral lineages to diverge from one another and acquire mutations. They have to get different. Then they need to transmit in the same area, right? So they have to get very different, but also not coalesce. So they remain different in these areas. They need to actually co-infect the exact same individual, and not just that, they need to be replicating simultaneously in the same cells of that individual. And then finally, I've got to find it. So it's got to come back out. Someone has to grab that sample and sequence it. And so the set of conditions that are required here is I think, fairly daunting. And so it's something that we didn't necessarily expect to find a ton of, but it's still important to look and think about it. And so most of the ways to get into the method of how we actually do it, most of the ways that do this they have tricks to prune their search space, basically. So one way I can do that is to say, I'm going to find the characteristic mutations that define, say, clade 20b2 and 19a4, and I'll just look for viruses that have some of each of those characteristic mutations, right? And so this works as long as I'm okay with only detecting recombinant lineages between these two lineages and not any of the, between 20b2 strains, something like that. Even though there might be a ton of genetic diversity within those groups I've labeled, I'm going to ignore it, but I'm just going to look at these edges. Another way you've seen people do it is just to use an absolutely tiny amount of samples and just look for cases that aren't very tree-like, and it does sort of work as far as I can tell, but it certainly won't be comprehensive, and it won't give us a very clear picture of all the viral recombination events that I might expect to see if I could use them all. Okay, and so that's where we got into. So it's again work mostly by Yatish and Brian, where we went back to that mutation annotated tree. We said this is actually a really, really great way to search for recombination as well, with some added features. You can't do it just core. And so what I'm doing here is I'm showing you a mutation annotated tree where each of these boxes is the position along my toy tiny viral genome where there's a mutation, right? So there's not many of them, and just to give you an idea of how this works. And so if I was to look at the multiple sequence alignment for these, it would be something like this. Again, where I'm sort of agglomerating mutations down from the root. So 10 is present in those 3 samples, 30 over here, etc. So you can kind of reconstruct the multiple sequence alignment from the mutation annotated tree. And then if I want to look for recombination, so maybe I think, well, branch F is long, right? There's 2 mutations on that one branch. That's not very common in SARS-CoV-2. Maybe it's special. One way to do this that we kind of came up with our simple algorithm is just to exhaustively split the genome and try to put them back, right? So I basically say, I'm going to split it here, and I'm going to put the red haplotype back wherever I think it goes, and the blue haplotype back wherever I think it goes. In this case, we see an impartial score improvement of 1, which ends up not being significant in simulations to calibrate this. But if we subdivide somewhere else in the genome, for example, right there, we actually get a really, really nice situation. Example F2 has no mutations that distinguish from node E, and F1, the other split, no mutations that distinguish from this internal node on the tree. And so that's a case where we think, okay, maybe recombination is a pretty good explanation for why it looks like there's sort of 2 different genealogies in the same viral genome. So that's the big idea here. I'll show you a little bit about how it works. So it's fast and it's sensitive. So it gets pretty good detection rate when we simulate these things at random. In red, I'm showing you the cases we don't get. And what's happening there is those are cases where the distance in starting parsimony score, so the distance between the parents, is relatively low. And it's actually quite hard to tell if those 2 genealogies are different or not, because they're actually very similar genealogies that may have given rise to the sample. As you get further and further out, it becomes very, very easy to find these things. And in fact, many people found them by eye early on in the pandemic. So it certainly can be done. Okay. Ripples is fast. So it's using a lot of the same tricks. Those of you interested in methods and computational development should know that if you can reuse code you've already spent optimizing, you don't have to optimize new code. The best trick we ever came up with here was if we cast this as a placement problem, we already kind of know how to do that. And so when we run this thing, it can search exhaustively the whole 16 million sample tree in less than an hour. We don't do exactly that, but we do have a daily updated version of this that will tell you every new sample that we've added, whether we think it may be recombinant or not. And the cool thing about this is it's real time, right? Because in the same way that Usher is, if I add a new sample to my tree, I can do that in seconds, I can then take that exact same data structure that comes out and exhaustively search the tree for recombination for that new sample in just a handful of minutes or seconds also. And so at the end of this whole thing, I'm able to very, very quickly tell my users, okay, I think that may be recombinant or not. And just to show you what some examples look like, so these are some, I picked out the cleanest ones, of course. These are some of the cleanest examples where what you can basically see in the middle in each case is the recombinant, and this is all the mutations that distinguish the putative parents, the acceptor and donor, top and bottom, from each other. What you can see is along a big stretch of the genome, the recombinant will match one parent, there's a break, and it matches the other for the rest of the way. And you actually see that a few times over here. So same sort of thing along the bottom here. And the far one has 2 recombination events. So we also infer those cases where you can see that it kind of switches from one to the other and then back. So again, consistent with the idea that we're seeing these nice recombination events here. Yeah, okay. And so when we do this, in aggregate, what we found was a little under 600 total recombination. This is back when the paper was published, so only 1.6 million samples in the tree. There are more today, but I haven't updated it since then. But of those, what that basically means is about 2.7% of all the samples had detectable recombinant ancestry. And so what that basically means is there's a recombination event that occurred, or a few of them relatively high up in the tree, such that they have many descendants that share that recombination event. If we use this to estimate sort of the classic parameter of microbial evolutionary genetics, it's called R over M. And what it's basically trying to capture is how much of the variation I see in a given sample is due to de novo mutation versus recombination, right? And when we do that, we get a number that's much, much less than 1%, suggesting that the vast majority of variation in SARS-CoV-2 is de novo mutation. Recombination is occurring, but it's relatively rare and maybe not the biggest impact that we're seeing. Okay. The other really cool biology you get out of this is if we cluster them all, we take them, and now I'm displaying sort of the confidence interval for each breakpoint as a gray line. There's 589 of them. And we just take a density function over the top of that, we can kind of see where there might be more evidence for recombination along the viral genome or less, right? And what we're seeing here is that actually the spike region, that's in blue on the edge there, has a pretty big excess of recombination events. As far as we can tell, it's about 3 times as much recombination as the rest of the genome. There are important detection biases, mostly the fact that the edges are very hard to detect because you might only get 1 mutation or something like that. So it's not a perfect picture, but it's certainly consistent with the idea that there is something special about the back third of the genome, and it's not just recombination. The mutation spectrum is slightly different and all sorts of other things as well. So we know something's going on there. It's also the place that all of us are staring to look for antigenically novel strains, because the spike region kind of is the main determinant of whether or not a new strain will behave differently than previous strains we've seen. So it's something we were really excited about. Of course, as I've said, this is rare. So the vast majority of those strains that have evolved without the assistance of recombination. And I don't think it's critical to this, but it is something to be aware of and look at. Okay. The other thing we really liked about this was, kind of while we were writing up our work, we became aware of this other work that was looking at very deep time, so super distantly related beta coronaviruses, and had a very different approach, but was estimating recombination rates on very fine scales. And they got a picture that is strikingly similar to our picture, right? It's not identical, but literally a 3 times higher recombination rate in the spike region, the sort of back third of the genome relative to the rest of it, suggesting this might be a pretty conserved process across beta coronaviruses with the same sort of genome structure organization that we're seeing here, which I think is also incredibly cool. Okay. Finally, just practically, it does matter for tree building. It has impacted the Pango lineage system. This is one of the examples we were able to find. There's actually quite a few others where the root of one lineage is inferred to be a recombinant between 2 other lineages. So it's actually, you're not tracking it exactly like you think you are. Your Pango system is supposed to represent your phylogeny, but of course it doesn't if your phylogeny isn't really a tree. Okay. All right, and we'll do this real quick. Some checks you might want to do. Is this any good? Well, you might ask, if my recombinant lineages have to be in the same place at the same time, do we see strong geographic overlap between the parents and strong temporal overlap between the parents? And we do, right? So relative to randomization tests, we see that there's quite a lot more recombinants in the same basic geographic region, with their parents in the same geographic region, and quite a lot more overlap in time than you'd expect by chance. That is to say that they were transmitting in the same human populations at the same times in huge excess of what we'd expect, strongly suggesting we're finding the kind of thing we're looking for. Okay. So to conclude from this section, so Ripples is the program's name. I didn't go into that. It's not important. It enables real-time recombination detection in newly sequenced SARS-CoV-2 samples. It's uncommon. So we do get recombination, not super common, 2.7% of all the viral genomes at the time, and actually much more now because some recombinant lineages got pretty big recently. So there's quite a lot now, but it's still not super common on a per transmission basis how frequently we see recombination events. And they are disproportionately common in the 3' portion of the SARS-CoV-2 genome. Okay, and at the end here, I will I'll do a little bit, I guess, of speculation about what I think of the future of online pathogen phylogenomics is going to look like, and I'll tell you folks what kinds of platforms and processes we're building to try to do the same sorts of things or enable the same kinds of analysis and real-time feedback for other pathogens as well. Okay. So the obvious thing, everyone asks me this every time I present this at various meetings. Someone comes up to me and says, "But I study this fungus, or I study MTB, and I want the same kinds of things." I say, "That's great. That will keep me in business because I'll build these things." And so that works for me. And so what we've been doing is spending some time going through the code base and trying to decode decovidify it. That is to say that there's a lot of assumptions we made when you're building this thing for a pandemic in real time, and you don't think about too hard. There were some simple ones, like for example, we hard-coded the name of the genome, and that's obviously not particularly useful. And some less simple ones that took some time to sort out. And so we've been spending quite a lot of time on this, but we've been able to, as a first test case, take this and look at Mycobacterium tuberculosis, so TB. The reason you want to do this is it has the same sorts of properties as SARS-CoV-2. You can think of it as an extremely slow-moving pandemic, but it is the #1 cause of death by respiratory illness in the world, briefly surpassed by SARS-CoV-2, and it has reclaimed the title. So it's out there. It is definitely a problem for humans, and it has a massive data science crisis. There are literally hundreds of thousands of genome sequences, and people's biggest trees so far have been about 2,000, and they're really struggling to handle it. So it's been a good case for us. And it's something that we're about to release kind of any day now. And so something we're really excited about getting out in the world. The other thing I think is going to happen is like more sophisticated phylogenomics platforms. So like the way we ended up doing this that I think will generally work is that I infer a tree, and then every day I do my sample addition. And I didn't talk about this much, but we actually also throw on what's called optimization. Right? So we're not just adding samples sequentially. We go back and we ask, can I rearrange this tree so I find a better ordering of my samples such that the tree is a little bit more optimal with respect to the mutations? And then just repeat, right? Like I just take the output of this and I throw it back into my process and the day starts again. So we've kind of built this thing. We think it works, but the major things it needs is more access to public health clinicians, to real-time genomic epi folks, all that sort of stuff. And so one of the things we're trying to do now is to make this a little bit easier, in particular to add what we call phylogenetic annotations, right? So the big annotation we have right now is the lineage system, and are there mutations that we know are important in this sample or lineage, right? I basically spit that information back to you. I say, hey, I see this spike mutation. You should look at this. Or that's one of this emerging lineage. You should look at this. But there's going to be so much more we're able to add because all sorts of things, all sorts of important traits evolve for pathogens on a tree, right? So for example, the big one we're looking at for TB is antimicrobial resistance type genes and sort of drug resistance genes. These are things that evolve super rapidly. We know they're out there, and we know those samples tend to be clustered on a tree because they're spreading very quickly out into the world because they get around our treatments. So they're really bad for us. And so that's one of the major things we're trying to do in terms of building toward what we think the future of this is going to look like, where a sick patient may come into my clinic. So this is an individual, and I won't even know what they have, right? There may be site tests for this, but for the next pandemic, there probably won't be. And so what we're going to do is take, full-on agnostic metagenomic samples. Just take everything they've got and figure out, sequence it and say, what's in here? As soon as I know this, I will add that to my global tree, right? And so I show this as a nanopore. I should have a conflict of interest statement that says that our department receives royalty funds from Nanopore, and I really want you all to use them and so we can have seminar series and other nice things. But they're also very nice because you can do them locally right in front of you in that moment and see the data straight off the machine. And so also Nanopore. But once I do that, I can take this sample, I'll figure out what it is, I'll add it to my tree, and I'll be able to digest this information, right? So the idea is that I'll tell, if it's a clinician, we'll figure out what's special about this sample relative to others, et cetera. And we'll say, okay, we'll give you a phylogenetic place. If you're public health, this will give you some clues about transmission in your community. And that might tell you how to take action to shut down the spread of this particular pathogen in your local area, whether you need mass mandates, et cetera. But also we'll give you things like these drug resistance mutations. We'll tell you, we think this strain is important because its close relatives are all resistant to the particular drug. And so that's one of the things we've worked really hard on with TB, is to go through all the literature and mine the sort of infinitely many various annotations and try to put them in there for people so they can interpret this. And it's not fully automated. Right now, I just tell you what I know. In the very near future, we're going to have tools that digest that, and these will be based on LLMs and other things that tell you what you want to know about this without me starting to spend some time stare at this and figure out what's important for you. And so that's where I think we're going to go. That's the future I'm trying to build, and I think many of us are trying to build. Just at the very end, here's some acknowledgements and thanks. The real people that did everything are basically Yatish, Angie, and Brian here. We've had a ton of great collaborators that did lots of different parts of this project. Some wonderful funding and friends that have contributed to this whole thing. And I think something I like to emphasize that I don't exactly know how to represent this on an acknowledgement slide, but the pandemic, this tool only works because the entire world is sequencing the virus genomes and sharing them with us as fast as they can, right? So and that is, for one lab to do that is just as hard for me to do this. So it's impossible to accurately and correctly represent the amount of labor that went into generating these kinds of data sets. And I should figure out how to include that in my acknowledgement slides, but the whole world has contributed. It's been incredible to be a part of this global effort to try to respond very quickly to this. And it's something that I hope we do going forward, is continue to share data this freely and this quickly, so that we can have a real-time idea of what's going on in the world. And anyway, we're seeing it somewhat with H5N1, maybe other things. Hopefully we learn this lesson and prevent the next pandemic. All right, that's what I've got. Thank you.